I am Pratyush Pandey from Educational Series. Today I have come up with a new topic, cell division, meiosis. So let's start this amazing video. Before this, I'll upload 3 videos regarding cell division part 1, cell part 1, cell part 2, then cell division part 1, and this is cell division part 2. So let's start. Meiosis. Now meiosis is the process when we use our human body use for making sex cells. For example, reproduction, for making small babies or embryo, to make for human civilization or for the human human species to evolve or to move on. This meiosis is very important. So how does this meiosis happens? What are its subphases and what happens? Each of this is what we learn in this video. So let's start. Meiosis is divided into 2 parts: meiosis 1, meiosis 2. And meiosis 1 there is prophase 1, metaphase 2, and metaphase 1, anaphase 1, and telophase 1. But in meiosis 2 it's another process. Just after meiosis 1 it starts meiosis 2, then prophase 2, metaphase 2, anaphase 2, and telophase 2. So now we can then this prophase 1 is further divided into 5 parts: leptotene, zygotene, pachytene, diplotin, and dikinesis. So here you can remember it by लटकती जुल्फों पे 2 दीवारें, or you can लटकती जुल्फों पे 2 दीवारें. leptotene, zygotene, pachytene, diplotin, and dikinesis. So now let's see what happens in each of them. Leptotene. Now prophase 1 is very vast in meiosis. So leptotene. Now let's learn more about this all in the next slide. Yeah, here we are. Let me zoom, and this all written part, if you want it, it's in the box below, description box below. So leptotene is the first part in what happens that the chromosomes, now which the daughter cell had, or that cell is there, the chromosomes, now the DNA is packed with the protein and the chromosomes begin to condense and solidify. In the next, zygotene. Now in what happens in zygotene, that they make pairs. What happens that suppose in, now after that things, there are a father, again some cells are from father, and some chromosomes are given from father, and some chromosomes are given from mother. So now for our embryo to develop, they both some kind of features such as eye color, nose color, hair color, and texture, color of the skin, and these all comes from both the parents, and such as for the eye color. So these both, for example, a father has red, black, and mother has blue. So they both genes come together and they make pairs. For they have the same purpose. Two chromosome, one from father, one from mother, make pairs. Then of hair color, then of the gender, and all of these come together. So now let's see this, and then zygotene and then come pachytene. In pachytene, what happens that these chromosomes pair. Now, how you may have thought that you are a sibling, has same father, mother, same parents, but how they are so different. You don't look like a sibling unless you are twins. You don't look like your sibling who is not a twin. He can have same kind of hair, same kind of face, but not exactly similar in all the facial expressions or the facial things, qualities. It's not same as a sibling, and that what causes diversity. The thing that causes diversity is that this process, pachytene in meiosis prophase 1. So a small thing can cause a big difference, and that causes diversity in human life or the human civilization, humans we can say. So pachytene, now for I and I, they cross over. Now it means that they exchange genes. They take some genes from this and from some gene from another, from gene from mother and then they cross over and they exchange genes. They give them to each other, and that genes, what happens, they call gene. They become gene. So this crossing over causes hybridness. As for example, your father has blue color and blue color eyes, and your mother has black color eyes. So when in this process packaging, when they cross over, then what happens that the genes are exchanged, and more they cross over, the more the meet at different angles. As you can see, this blue part is meeting the green pink hair. No, only here they are meeting. So more the places they meet, there will be more mix. They all some things from some ingredients from father, some ingredients of mother, and they mix and mix and mix, and more they mix, the better. The better is the both thing better. The result is the more they mix. So it depends on case to case. It's enormous. It does not depend on anything. That's its luck and how much they do it, and then they got this crossing over finishes, and then what happened that this embryo is blue. It's suppose they are blue color, some part gets pink, and pink that it parts get blue, which causes diversity in siblings. As now when the egg cell and the sperm cell, yes, the sperm cell will meet, then the embryo, and another time when the egg cell and sperm cell will meet. So then that variety in that forming can cause different facial expressions, so the qualities of or physical things in the two siblings. So now packaging is done, and we come diploid. Now as they were joined at many parts, and chlamydomonas, or we can say, I'll help you. You can see here, here, yeah, chiasmata. I remembered it. Next I wanted to show you the spelling. Chiasmata. The joint places wherever they join, these are called chiasmatas. So the chiasmatas get against, as you can see, they go away. So now let's see dikinesis. In the dikinesis, what happens that all the chiasmata or chiasmatas points get away, and the sister chromatids go away, and they just roam randomly till the prophase 1 ends and the metaphase 1 start. And as we know, the spindle fiber, the nucleus disappears, and the spindle fibers are ready at their position, clapping for them. Now let's say and enter metaphase 1. Let's appreciate it's coming. Welcome metaphase 1. So now what happens that these now in earlier what happened that in mitosis they became in a single file line, but here they arrange in pairs. It's not of this, it's showing something else, but what it happens, they do not arrange in a single file line. In GF showing wrong, just they get in the pairs, and the spindle fibers attached to the centromeres of the different pairs. They just don't go away from each other, or they don't even stick. They just be here, and they're not attached but close to each other. These two chromosomes which have exchanged their genes, and then the spindle fibers connect each other like they hold of the anothers, and they became in a paired line. And this ends metaphase 1 as they get arranged. The spindle fibers adjust, some get short, some get long, to adjust them in the middle. Now that's one main important thing that which we did, that by cross section in that prophase 1, we call, sometimes scientists call that scientific, we can say scientific name, they call that structures homologous structures, these complex structures which these form during that third part. You write that all. Now I have got you a homework. Write all that the parts of prophase in the comment box below, and write all in the order. Don't forget to write them in the order. So now where we were, anaphase. Now in earlier what happened that the sister chromatids were pulled away, but here now this is also wrong. The sister chromosomes are pulled away. So when the side all the things get together, and the sister chromosomes are pulled away. Unless what happened in mitosis, that they were sister chromatids. Here are sister chromosomes. Telophase 1. So now what happens in telophase 1, thus the nucleus appears, and the two nucleus and all the cell organelles appear, and not now here cytokinesis is not another part, it's just there, and in the telophase the cytokinesis does, and we get two daughter cells which have 23 and 23 cells, 23 and 23 parts, 23 and 23 23 and 23 chromosomes. Earlier it was 46, now they divided 26 into 23 and 23. They don't didn't duplicate, just they it halved, and two daughter cells are 23 and 23. Now prophase 2. What happens in prophase 2, that these spindle again they become 23 chromosomes. This resembles normal meiosis. Then that nucleus disappears, and then again the meiosis, this mitosis similar process starts, and then came this prophase 2 ends. Then come the metaphase, where now the same as mitosis, sister they come in a single file line, these chromosomes and the spindle fiber attached to the sister chromatids, and they arrange in the middle. Anaphase, A for away, the sister chromatids are pulled away. So our centromere spills, and these are pulled away. Telophase 1, same thing, the nucleus appears and the all cell organs appear, and thus they become two till the cytokinesis separates them both. Now you will say that it will all even get half of it. No, it is what it gets. Now its parent cell, the DNA was replicated, then they were 4, so you can say 2 and 2, and then they again divided, so they get 1 1 and 1 1 1 1 and 1 1. They got into 4 daughter cells. Now because we learned that this does the metaphase 2, meiosis 2, and this got into 2, and this also did another daughter cell, they also did the same, and at the result we get 4 daughter cells, each with 23 23 23. As when it duplicated, it got 48 plus 92, then it got 48, or we can say 23, and then they become 23 23, or the result are 23 23. And these do not duplicate, just they are called gamete cells or the sac cells. And remember, in males it makes sperms, while in females it makes egg cells. Sperm cells are relatively small compared to the egg cells. Now significance of mitosis. Now you have learned about mitosis, that it duplicates, it runs and multiplies the cell, the same kind of cells. So it helps in growth. If your hand wants to grow and you are growing tall, suppose I have also grown tall. No, I have raised. So that if you growth of your hand, growth of your body, and growth of your skin, damaged wounds, and this all when you need the circumstances, when you need the same kind of cells to replenish those who are wounded or those who are dead. So there comes the help of mitosis, and this you can see the stage of mitosis. Just a revision, I had showed you this GIF at the starting also, earlier video. And the GIF, you can see cytokinesis, and this is the mitosis. Now let's see significance of meiosis. Significance of meiosis. Now for the human generation, now suppose your father and your mother, and when you were born, that if you had not been born, then if no ever children will be born, or no this meiosis would occur. So that will it happen, that will humanity go ahead, because when your parents will die, then there will be no offspring. If there is no offspring, then who will take the human generation? How will the human generation proceed on? That species will go, and so that where reproduction happens. And how reproduction happens is meiosis. So meiosis is very important for the continuous going on of human species. And the results is gametes, in which now the gametes are 23 chromosomes, and they both meet and they become 46 chromosomes, and the embryo is then born. Then the result of meiosis is that embryo, and then it does meiosis is that embryo, and then it does mitosis many times till it grows into a human being. Now you see this, it's a very good animated GIF. Let's first see this. Now in mitosis is the attack of clones. Oh, I'm the king of clones. Attack clones. This is the parent king of clones. Now it divides into two small, and they both have, it had 46, means it replicated, it became 92, and then it got 46 and 46. 46 and 46, same type, you can see color, chromosome, same. While what happened in the meiosis, it's there to be different. It comes, then they occur to do different daughter cells with 46 and 46 added to 92, and then with 23. It's a little different of what we have learnt. Just 20, the result you can see 23, and these all are gametes, and any one of these who will be successful will go meet the egg cell, and then the embryo will be developed. You can see this GIF in the PPT. So my friends, this was all in this video. If you like my videos, please like, share, subscribe my channel, and don't forget to click the bell icon segment below to get regular dates of my videos. Don't forget to click the bell icon segment and see all the videos of this topic and all the videos of the channel. And stay curious.